Okay, picture a really intense, packed mosh pit. But instead of people, this mosh pit is made entirely of biological cells, jammed tightly together into living tissue. How do these cells manage to move around, rearrange themselves, and basically navigate their environment? And taking it a step further, what if that mosh pit isn't on a perfectly flat floor? What if it's wrapping around a sphere or stretched across some weird egg-shaped landscape? Welcome to This Explainer. Today, we're diving into the geometry of cell migration using what computational biophysicists call the vertex model. We're going to see exactly how an actual 3D terrain dictates the strict rules of biological movement. Here's a quick look at our roadmap for today. We'll start with the basic rules of cell migration, then move to life on a flat plane. After that, we'll throw a spherical curveball into the mix, explore the ellipsoidal extremes, and finally, wrap up with exactly why tissue geometry matters so much.
All right, part one, the rules of cell migration. Let's get our baseline concepts locked in.
So let's kick things off with a pretty fundamental question. What actually happens to a dense, gridlock traffic jam of migrating cells when the surface they're moving on is suddenly no longer flat? It's a super profound problem, right? Because in biology, real tissues aren't just flat 2D sheets sitting perfectly in a petri dish. They form intricate organs, complex tubes, massive 3D structures. And that geometry creates a very real physical hurdle for these biological systems to overcome.
To understand how cells actually solve this, we first need to nail down a key concept, fluidization. This is essentially a jamming to unjamming transition. Think of a highway packed bumper to bumper. That's your jam state, a rigid, highly ordered cellular structure. But as activity ramps up, the tissue unjams. It physically breaks down into this swirling, fluid-like state where cells can suddenly slide right past one another. So throughout this explainer, when we talk about a system going from low to high activity, this fluidization process is exactly what we're watching for.
Moving on to part two, life on a flat plane. This is our zero curvature baseline.
On a perfectly flat surface, the contrast between our two states of cellular activity is, honestly, night and day. In a state of low activity, the system behaves just like an ordered lattice. It's this pristine, intact honeycomb structure. The cells barely move, just kind of quietly vibrating in place. But crank things up to high activity, and that active motility totally shatters the crystalline order. This is what triggers the fluidization we just mentioned. You start seeing frequent neighbor exchanges, what the pros call T1 transitions, along with severe shape distortions and these massive swirling vortex-like patterns.
Visually, the transition is absolutely striking. When looking at a low-activity state, everything is a pristine, jammed honeycomb lattice. It's totally orderly, completely locked in place. But then, as high activity kicks in, that order goes completely out the window. The cell shapes distort wildly, the structure unjams, and you start seeing these intermittent gaps or defects tearing through the tissue as the cells violently push past each other.
We can actually measure this chaos just by looking at the cell speed. When cells are in a low-activity state, velocity vectors show them merely vibrating in place, just incredibly slow speeds. But in the high-activity state, boom, you get these massive, swirling vortex velocity patterns with massively elevated cell speeds. Now here is the absolutely crucial point to remember. Because this flat plane has zero curvature, this fluidization is perfectly uniform. It's spatially isotropic. That means the chaos is distributed completely equally across the entire domain.
All right, part three, the spherical curveball. Let's see what happens when we add 3D curvature into the mix.
Behold, the sphere. A sphere has what mathematicians call constant positive Gaussian curvature. And, as it turns out, this specific geometry fundamentally breaks the rules of our flat grid. Think about it. You literally cannot wrap perfectly flat hexagons around a ball without leaving gaps. It's mathematically impossible. The underlying geometry of the sphere actually forces exactly 12 pentagons into our otherwise perfect hexagonal lattice.
Now these forced pentagons actually have a formal name in biophysics, topological defects, or disclinations. They are structural irregularities that simply have to exist because the underlying geometric curvature demands it. It's kind of like a traditional soccer ball, right? It's mostly made of hexagons, but it absolutely requires those pentagons to actually close up into a 3D sphere. And in our vertex model of cells, these defects are way more than just neat visual anomalies. They actually hold tremendous mechanical power.
So, why do these 12 specific defects matter so much? Well, it's because they act as stress concentration hotspots. The tissue is literally stretching and pulling uncomfortably around these pentagonal zones. And because of that intense stress, they drastically increase the rate of those T1 transitions, the neighbor exchanges we talked about earlier. This means that when the cellular tissue starts to unjam, it doesn't happen uniformly anymore. These defects act as the literal epicenters, initiating local fluidization much earlier than in the perfectly hexagonal zones.
Looking at the velocity fields brilliantly illustrates this localized effect. When the sphere is in a low-activity state, it's calm, just vibrating uniformly, no big deal. But check out the high-activity state. Those fast, high-speed velocity vectors, they aren't spread out evenly like they were on the flat plane, not at all. They are concentrating around very specific zones. The spherical Kern ball has essentially created isolated hotspots of cellular chaos exactly where our topological defects are located.
Let's step it up a notch in Part 4. The ellipsoidal extremes. What happens when curvature becomes non-uniform?
So the rules are about to change yet again. Unlike a perfect sphere, which curves exactly the same way everywhere, an ellipsoid has non-uniform positive Gaussian curvature. It maxes out at the extreme tips, the poles, where the curve is the sharpest, and then it minimizes along the middle, the equator, where it's relatively flat. And because the curvature is no longer uniform, those topological defects, our structural pentagons, start behaving really differently. In a low activity state, the defects are relatively stationary on the surface. But when activity ramps up, these defects actually physically migrate. They literally move and segregate towards specific regions to minimize their elastic strain energy. They're basically trying to find the most comfortable geometric spot on the entire landscape.
And where exactly do they go? They migrate straight to the high curvature zones, the poles of the ellipsoid. And because all the defects cluster at the poles, the active fluidization becomes strictly non-uniform. The geometry of the surface is quite literally steering the biology. It's drawing the points of stress to the extreme ends and creating highly uneven zones of fluidization. So now, the cell speed distributions are directly modulated by the varying curvature profile underneath them.
Taking a look at the velocity fields absolutely proves this point. You've got minimal movement in low activity, but during high activity, notice exactly where those high-speed velocity vectors are clustered. They aren't random. They are heavily concentrated right near the highly curved poles where all those defects gathered up, leaving the flatter equator region relatively calm. This conclusively shows us that cell migration efficiency is locally controlled by the specific curvature right beneath it.
Finally, Part 5. Why tissue geometry matters. Let's synthesize all these dimensions. This summary perfectly wraps up our whole journey today. On a flat plane with zero curvature, the jamming to unjamming transition is completely isotropic and perfectly uniform. But add constant positive curvature with a sphere, and suddenly the underlying math forces 12 structural defects into existence, which creates these localized fluidization hotspots. Stretch that sphere out into an ellipsoid with non-uniform positive curvature, and those defects physically migrate. They cause intense, non-uniform fluidization specifically at the high curvature poles. Essentially, the geometry dictates the rules of the cellular mosh pit.
So if there is one crucial takeaway I want you to remember from this explainer, it's this exact phrase. Migration efficiency is locally modulated by the varying underlying curvature profile. The literal shape of the floor determines exactly how and where the cells can move. It is an absolutely incredible dance between physics, math, and biology. Which leaves us with a pretty thrilling question to ponder. If the underlying mathematical curvature ultimately dictates exactly where cells jam, unjam, and migrate, what other biological mysteries could vertex models help us unlock? Are we talking organ development, tumor metastasis, wound healing? The shape of life is quite literally driving the mechanics of life. Thank you so much for joining me on this explainer. Stay curious and keep looking at the incredible structure behind the science.